Okay, so hello everyone. I'm Mikołaj Kuska, and I'm so happy to welcome you to the next seminar of the Polish Astrobiology Society. And today's speaker is gonna be Rosa Santomartino from the UK Centre for Astrobiology at the University of Edinburgh. And the title of her lecture is the Space Biorecycling, How Can Biotechnologies Support Space Sustainability? So welcome you, Rosa, and the floor is yours.
Thank you so much. I'm going to share the screen, of course, so that you can see my presentation, hopefully. So thank you very much for having me here today on, I was saying, on a Sunday afternoon. My name is Rosa Santomartino, and I am a researcher at the University of Edinburgh in the United Kingdom. I'm one of the co-director of the UK Centre for Astrobiology and a Leverhulme Research Fellow, and also a principal investigator of the Space Microbiology Group. So what I'm going to talk about today is space biorecycling. So as you can read from the title, how can biotechnology support space sustainability, or in other words, space exploration in a sustainable way? We will see what does this mean. So what I'm going to talk about today is what I focus most my research, as you may guess from the title of my group, which is Space Microbiology, is of course microorganisms, so microbes. I'm not sure about everyone's background here, so I'm going to give you a brief introduction of what microorganisms are and why they are important. And then also which are the space conditions that are relevant from the biological perspective, something about the goals for human space exploration in the future, and then the roles that microorganisms could play in this. Finally, I'm going to talk about some of the experiments that I performed in the past, which focus mostly on an application, a biotechnology called biomining, applied to space, so space biomining. And then eventually I'm going to tell you something about biotechnologies and space sustainability. And finally, I will leave you with some take-home message and something about my career in astrobiology, just because I thought that this may be useful for some people here that may be early career scientists that maybe want to get into astrobiology but are not sure how to. So first things first, what is a microorganism? Sometimes we refer them as microbes, sometimes we refer them as germs, although this is not scientifically accurate as a term, but let's explain what they are. So this representation that you can see here, it's a graphic representation that biologists use when we want to classify all living organisms, and it's called the evolutionary tree of life. And this is, again, a classification of all the living organisms that we are aware of, that we discovered, that we know about. And when we talk about microorganisms, we are referring to, let me see if I can take a laser point. Here it is. We are referring to bacteria, of course, archaea, but also some eukarya, like, for instance, unicellular fungi. In the everyday life, you will find unicellular fungi in the form of molds. Like, I don't know if you ever had these horrible molds forming in your shower or in the toilet or something like that. That is a unicellular microorganism called fungus, is a fungus, basically. So as you can see from this representation here, microorganisms are very, very diverse in forms, shapes, phylogeneity, metabolomics, a lot of things, metabolism, and a lot of different characteristics. And they can be, for instance, unicellular algae, archaea, bacteria, fungi, as you can see here, viruses, protozoan, and so on. So as you can see, they are all quite diverse one to each other, but eventually they have some characteristics in common that make them all microorganisms. The first characteristic that they have in common is that they are very small, in the orders of micrometers. A micrometer is 0.001 millimeters, so you can imagine how small they are. And this is why they are called microorganisms. The second one is that they are unicellular organisms, or meaning that they are made of a single cell, or in some cases even less, like for instance in the case of viruses. And finally, they have been found almost everywhere where we search for on Earth. on Earth. I want to highlight on Earth because it's important from the astrobiological perspective. When I see that on Earth we found them almost everywhere, I mean literally almost everywhere. In fact, they have been found on soil and rocks. They have been found on pipes, devices that have been found on buildings, infrastructures, and so on, but also in some conditions that we call extreme conditions, because they will be extreme from our perspective, which is, for instance, very hot environment, very cold environment, very dry, very salty, a very high altitude, like for instance, microorganisms that we found also on clouds. And last but not least, they are also present on other living organisms, including animal, plants, and of course, ourselves, so human bodies. We are animals, so of course, they are also on us. So this tells us that microorganisms are quite versatile and they can survive to extreme environments, a variety of different environments. But, we are talking about astrobiology. So if I ask you which is the harshest environment I can think of, I don't know if you agree with me, but to me, the answer is space, of course. And why do I think that space is one of the harshest environments that we can think of? Well, basically because of the conditions that are present in space, which have some relevance from the biological perspective. Some of these include the fact that gravity condition in space is generally different than those on Earth. Generally speaking, we have a gravity condition which is lower compared to Earth. For instance, we are thinking about open space or orbiting around Earth, then we are going to have a condition of gravity which is called microgravity, which more frequently you will hear weightlessness or absence of gravity. From the physical perspective, this is not really correct, but we understand what it means. It's a gravity condition which is very, very close to zero, and this is why it's called microgravity. But if you go on Moon or Mars, you will have a gravity condition that is not close to zero, but it's still lower compared to Earth. For instance, on the Moon, you have 0.16 G, while on Mars, you have 0.4 G. So still a gravity condition that is less than open space, but also lower than Earth. But then in space, you also have a higher doses of radiations, which is one of the most important issues to address when you think about human space exploration. Because if we think about going to Mars, for instance, this means that we are going to so a travel to Mars takes at least 7 months. One of the major issues for this is that you are going to expose a human or a living organism anyway, for 6-7 months to a higher doses of radiation. So this is going to be very deleterious for the health of the human that is going to Mars. And this is something that a lot of space agencies are trying to solve in one way or another. But the doses of radiation, the type of radiation that you will experience in space are generally higher and of different types compared to those on Earth, because on Earth we have the atmosphere that protects us from some of them. Which are these radiation types? Galactic cosmic radiation, solar cosmic radiation, Van Allen radiation belts, and solar electromagnetic radiation. I said just a second ago that is the atmosphere that protects us from the radiation. It's not just that, it's also the magnetic field of Earth. And then what else do we have in space? Well, in the majority of open space, we have a condition of vacuum. Vacuum is a condition of very low pressure or absence of matter. Again, from the physical perspective, there is no such a thing as absolute absence of matter, but the pressure can be very low, so low that it can be very, very close to 0 Pascal, as you can see from these numbers here. And finally, another extreme that you will find in space is, of course, the extremes of temperatures. It can get to as close to the absolute zero as high as billions of degrees if you go closer to the sun, for instance. But even if you just focus around the low Earth orbit, which is the orbit that surrounds Earth, the temperatures are still quite extreme, because they can go to as low as minus 157 degrees Celsius to as high as 121 Celsius degrees. So still you have these extremes of temperatures that we ourselves won't survive to. And if you think about a planetary body, like for instance, Moon or Mars, then there are even other conditions that you have to take into account. For instance, you have to consider the presence of rock and regolith, and in some cases even dust. These rocks, regolith and dust, can contain by themselves some toxic compounds. They can contain toxic salts. Can be the dust itself, the physics of the dust can be toxic for our lungs, for instance. And even from the perspective of obtaining, we will focus about this a little bit later, but from the perspective of going on another planet and finding food or resources, well, these rocks and regolith are generally poor on nutrients. So we have to consider this. And we also have to consider the atmospheric composition, which is going to be different than those on Earth, and the pressure that is going to be different. So the situation becomes even more complex. Despite all of these difficulties and the harsh environment that are present in space, still the goal for human space exploration is moving farther and farther from Earth. In fact, in the recent years, in the last decades, all the human space exploration has been focused around Earth orbit. Think about the International Space Station. The International Space Station is orbiting Earth in what is called low Earth orbit, which I just mentioned a minute ago. But the goal for the future is to move farther and farther from this, for instance, reach the moon first, to then form and establish constant human presence there, and then move toward Mars and beyond. But as we think of moving farther and farther from Earth, we have to think of the resources that we are going to need for this long-term and long-distance human space exploration. The resources that we are going to need include, for instance, things like food, water, oxygen, which are basically life support systems, resources that are going to sustain our own survival. But then eventually, in the long term, we are going to need shielding material, something that gives us energy, something that gives us materials, and so on. And then even in the longest term, we are going to need probably long-term necessities. Think about pharmaceutical compounds, for instance. We can bring as many pharmaceutical compounds, as many drugs with us for the travel, but then the space conditions may make them expire earlier than usual, and eventually we are going to need them in the long term. So in the long term, we are going to need even more resources. Now, if you think about let's just talk in terms of money, right? So at the moment, the cost of launching a single kilograms of any material to space can cost as high as almost $20,000. Now, if you consider that the trip to Mars is, as I said before, at least 7 months, and then you have the time of the surface mission, and maybe you have the return mission time, so other 7 months, you can imagine that you cannot possibly bring all these resources that you are going to need for 7 months, for 7 plus 7 plus surface missions time, just with you. Imagine if I tell you, okay, you have to go in a trip to I don't know, the Sahara Desert. You can never stop to, I don't know, a shop or anything to bring food. You have to bring everything that you need with you. It's going to be an enormous amount of stuff that you are going to bring with you, and it's also an enormous cost. What does it mean? It means that if we want to achieve deep space human exploration, we are going to need ways of obtaining the resources, the thing that we are going to need in the place in which we are going to settle, to move, and to travel to. This is a concept that is normally referred to as in situ resource utilization, which is basically just what I just said. So the technologies that allows us to obtain the materials that we are going to need, the resources that we are going to need in the place in which we are going to settle, rather than relying on a constant resupply from Earth that, as you can see, is not sustainable in the long term and in the long distance. The good news, from my perspective as a space microbiologist, is that microorganisms can really help. And why do I say that? Well, first of all, this gives me the reason why, to me, is interesting and important to study microorganisms, study space, and study the two things together. So microorganisms in space. Microorganisms are resilient. They are model organisms. There are implications for human health in studying microbes. They are important in biomanufacturing and bioindustries. they, in general, biotechnologies that utilize microorganisms require less energy and less resources. They are essential to create habitable environment, and also, generally speaking, the technologies that rely on them are sustainable. And why do we want to study them in space? Well, because as we've seen, microorganisms can survive in a variety of extreme conditions, and we've seen how much space is an extreme environment. They are important for human space exploration, but also just because, why not? It's very interesting to see how these living organisms can thrive and survive in such a harsh environment as space. But there is another reason why we want to study microorganisms in space, and is that, if you remember, I mentioned before that microorganisms are also on our own human body. They are everywhere. So if we are going to go anywhere in space, literally anywhere, we are going to bring our microorganisms with us. So it's going to be impossible not to bring them with us. And so it's going to be quite trivial, quite important and non-trivial, what I wanted to say, to understand how they behave under space conditions. Now, why do I think the microorganisms will be absolutely necessary and important for us for space exploration? Well, because in our everyday life here on Earth, they are already performing a lot of different roles for us. Think about how much they are used in the food industry, in the pharmaceutical industry, they are important in the production of fertile soil. They are important in some biotechnologies, like, for instance, biomining, which I'm going to mention more in detail in a second. They are important for our own health. And last but not least, they are important in the recycling of waste, so from a sustainability perspective. So because they perform so many different tasks and roles for us on Earth, imagine how many of these roles could be actually translated to a space environment, to space exploration. And to make some examples of this, microorganisms beyond the Earth, on an extraterrestrial settlement, for instance, can help us generating food and oxygen, can help us recycling the waste, can help us obtaining materials, and eventually also in some bioindustries and some biotechnologies. All of these tasks that you can see here are going to some way support one of these necessities of long-term space exploration, and they are going to sustain life support system and in situ resource utilization. Now, I want to focus on this particular technology here. This particular technology is called biomining, and I'm going to focus on this because you will see in a second that this is what my past feature research was focusing on. So what is biomining, which is also called bioliching sometimes? It's the process of using microbes to extract elements and metals of economic interest from rock ores or mine waste, as you can see here from the AGI definition. You can use both chemoeutotrophic and organotrophic microorganisms. In other words, there are a lot of different types of microorganisms that can perform biomining. And I know that this sounds a little sci-fi sometimes, but I have to tell you that nowadays, this technology is already used on Earth. In fact, roughly 20-25% of copper, 5% of gold, as well as other elements and metals are currently extracted on Earth using this technology. There are some advantages of using this technology compared to other, to traditional mining technologies, because not only it is complementary and it improves the rates of metal recovery from mine waste and dump heaps and so on, but it's also generally quite more economic compared to traditional mining and also more environmentally friendly. Now, because as we've seen, biomining is one technique that allows us to obtain metals, elements from rock ores and mine waste. And we mentioned how important this is going to be in space. You can imagine why there is been an increasing interest toward the possibility of biomining, of performing biomining in space. It's, of course, from the perspective of in situ resource utilization. Why does it matter? Well, actually, which are the planetary bodies that are of interest from the space biomining perspective? Well, definitely the Moon and Mars, because as we've seen before, these are the goal for the future human space exploration. But also some asteroids. For instance, some asteroids in the asteroids belt, which is the belt containing asteroids which is between Mars and Jupiter, and also some others closer to Earth, have been there are some data showing that some asteroids contain a lot of precious elements, like for instance platinum group elements and so on. So there is also an industrial interest in trying to understand if we can perform biomining to extract elements from the Moon, Mars, and asteroids. However, these planetary bodies contain, of course, have some conditions that are different from Earth. To mention one, gravity condition is different on these planetary bodies than on Earth, and is generally lower compared to Earth. For instance, we mentioned this already. The Moon has a gravity of 0.16 g, Mars is a gravity of 0.38 g, and asteroid has a gravity condition that is we can consider it to be microgravity or centigravity for the larger asteroids. So we want to understand if we can perform biomining in space. Okay, fair enough. But the question that we have to address first is, one, how does gravity influence the way in which the microorganisms are going to interact with the mineral, with the rocks under space conditions, under microgravity conditions? And second, does this affect the capacity of microorganisms to extract elements from rocks? So in other words, is the influence of gravity going to influence the biomining capacity of these microorganisms? There is not much in literature from this perspective, but with this idea in mind, trying to address these questions in mind, we performed two biomining experiments on board the International Space Station. One called BioRock, which was launched on board the International Space Station in 2019, and the second one called BioAsteroid, which is a little more recent, is being launched two years ago. So let's talk about BioRock first, just because it's the one that we performed before. With BioRock, we wanted to understand how three different bacterial species, which are Sphingomonas dissecabilis, Bacillus subtilis, and Cupriavidus metallidurans. Sphingomonas dissecabilis provided by our group in Edinburgh, Bacillus subtilis provided by our collaborators at the German Aerospace Center, and Cupriavidus metallidurans provided by our collaborators at the SEK Centrum in Belgium. Sorry, what did I say? DLR in Germany and this one in Belgium. And these three bacterial species were inoculated on a rock surface. This rock surface is a basalt taken from Iceland. Why did we select basalt? Well, basically because the Moon and Mars are rich in basalt rocks. So this was the first reason to try and understand if they could perform biomining on basalt rocks. But the second one is that basalt is quite a porous rock, so it provided a good support for the bacterial colonization. But of course, in order to understand if any effect of the extraction of element was actually due to the microorganisms and not just some chemical reaction given by the liquid, we also had what is called non-biological control, which was basically just the rock together with the liquid media and no bacteria. How did we prepare the sample? Basically, we had these pieces of basalt slide coming from Iceland that I mentioned before, and each hardware, we developed this hardware called biomining reactor, which was actually developed in collaboration with the Kaiser Italia. And as you can see, this hardware here can allocate two different samples. And here you can see one without any samples in it. And we were lucky enough to perform this experiment, because this experiment was launched on board the International Space Station. We were lucky enough to have the opportunity to go to the NASA Kennedy Space Center in the US in July 2019 to prepare the experiments. Here you can see an image of me, Professor Charles Coquel, and Dr. Annemiek Waken, which was a PhD student in our laboratory. And here we are very happy because we just just concluded the preparation of all the samples. Here you can see me as well, very, very tired, I can tell you, but also very happy that everything went okay from the perspective of the preparation. And here you can see, again, charts handling the samples to the International Space Station processing facility. And finally, here you can see part of the BioRock team happily going around the NASA Kennedy Space Center after completing the preparation of the samples. And so between that and the actual rocket launch. So again, we were very lucky. We had the opportunity to attend the rocket launch. The experiment was launched on board the space station thanks to the SpaceX Commercial Resupply Service 18, which was launched in July 2019. And here you can see, well, this is me on the rocket launch site, but here you can see it was quite far, to be honest. You can see here like a zoom of the picture. It was far, but it was still very cool to see your samples going in space. And here you can see a closer image of that. And then this is the capsule reaching the International Space Station. And here you can see the astronaut Luca Parmitano that is installing the hardware that you've seen before on board the International Space Station inside the incubators. These are European Space Agency incubator called Cubic that are present permanently on board the International Space Station. How was the experiment designed? Well, as I mentioned, we had these 3 bacterial species plus non-biological control. They were prepared in 4 different sets of samples. 3 sets of samples, identical set of samples, were launched on board the International Space Station, and they were subjected to either microgravity or a simulation of Martian gravity or a simulation to terrestrial gravity. How can you perform the simulation of gravity condition? Well, because the cubic incubator that I just showed you before has this little centrifuge here that by centrifugation allows to simulate gravity condition other than microgravity. But we also had a 4th set of samples that just stayed on Earth. We call this ground control, generally speaking. And it was just an extraterrestrial gravity control, basically. The experiment has been going on board, it's been running either on the International Space Station or on Earth for 21 days, so 2 weeks, and then they came back to Earth. Here you can see, again, images of the astronaut Luca Parmitano performing the post-growth image acquisition on board the International Space Station. And these are some of the images that he took. It was quite interesting because these images did not provide any really any quantitative data, but they provide us some qualitative data. For instance, it allowed us to see that there are some aggregates and gas inclusions that were present in different shape and different numbers depending on the gravity condition. And all of these features here that were possible to observe thanks to this photo session were not available anymore to be observed when samples came back to Earth. And these are just some images showing the samples coming back to Earth for us to analyze them on Earth, of course. Now, what about the results? Well, all the experiment had the main focus of understanding if space biomining could be performed under different gravity conditions. And so we focused on 2 different types of metals. The first is a group of metals called rare earth elements, which are quite important in every electronic device. And a second set of, a second element that we focused on is vanadium, which has a strong industrial interest because it's normally used in a structural material. So it's quite a sturdy and resistant metal. And what did we discover from this experiment? Well, we discovered that at least one of the bacterial species that we launched in space were able to extract rare earth elements and vanadium. These species were Sphingomonas dissecabilis and Bacillus subtilis under all the gravity conditions that we tested. So this was a very good news for us because it gave us an indication that the space biomining is in principle achievable under a range of gravity conditions. Of course, this is not a scaled up experiment. It was just to understand which were the mechanisms underlying, which were the effects of gravity conditions on the mineral interaction. If we want to know if this is something that we can actually industrialize, it's going to be more research is needed. But that was quite it was the first demonstration of this under the International Space Station. So quite interesting. And because this experiment went so well, then we decided to perform the second space biomining experiment called BioAsteroid. This time we decided to expand a little bit our knowledge of about microorganisms, different type of microorganisms. So what did we use? We used a bacterium again. This guy here, the Bacterium Sphingomonas dissecabilis, was also used in BioRock in the previous experiment. But this time we also decided to go further and try and understand if we can use the fungus, so another type of microorganisms, particularly the fungus Penicillium simplicissimum. And also a combination of the two, because we want some, in nature, you will very, very rarely find a single bacterial species or microbial species alone. Normally, you will find them in combination in what is called the consortium. So we wanted to see if the consortium was enhancing or hindering their capacity for performing biomining. And we also decided to change the rock substrate that we used. So if you remember, with BioRock, we use a terrestrial rock, the basalt rock coming from Iceland. This time we said, you know what, let's try with the real extraterrestrial rock material. So we decided to use crushed meteorite this time instead. The crushed meteorite that we used is a type of meteorite called L-chondrite. You can see here an image of that, and you may recognize the same bioreactor used for BioRock, but this time, instead of having the basalt slide, we had the crushed meteorite allocated here in the two sample sections. The experiment was very, very similar to BioRock, but a little smaller. In fact, this time we only had a set of samples launched on board the International Space Station as subjected to microgravity. And then we had a second of course, set of samples that stayed on Earth, and this was subjected to real terrestrial gravity. Again, this is our ground control. The rest was quite similar, also in terms of days, and the media use was the same, the temperature use was the same. So quite similar. And this time, the launch occurred in December 2020, which, as we all know, was during the pandemic time. So this time, unfortunately, we couldn't do all the way to the Kennedy Space Center, but I cannot complain, of course, because I have the opportunity to run another space experiment on board the International Space Station. But this time I had to attend the rocket launch from my computer here in Edinburgh, so from the UK. What are the results so far? The paper out of it is in preparation, but I can show you some of the results out of this experiment. The first thing that we tested was, okay, were the microorganisms able to grow on top of the meteorite rock in space, so under the microgravity condition? And the results, to summarize, the result is yes, they can. In fact, if you observe the bacterial species here, you can see that the biofilm, so there is a film here, a bacterial film called biofilm, growing on top of the rock surface. This image here are scanning electron microscope. Is a particular type of microscope that allows you to have these beautiful 3D-like images. And you can see here, you may recognize here some tiny cellular cell shape. And then when we observe the samples that contain the fungus, so the fungus Penicillium simplicissimum, this is what we found. Basically, these filaments here is called mycelium, and this is the way in which filamentous fungi tends to grow. And these results here just show you that, yes, the microorganisms, both type of microorganisms, were able to grow on the meteorite in space. So this is good news for us. The second thing, of course, was, okay, what about biomine? Did they manage to extract any elements from the meteorite rock under microgravity and Earth gravity condition? And the result is yes, they can, but it's quite, as you can see here from these two elements, here is phosphorus and here is vanadium. You can see that in green, are the samples coming from the International Space Station, so the space samples. And in orange is the samples from the ground control, so experiments in microgravity and experiments under terrestrial gravity. You can see that there is a difference between the phosphorus and the vanadium, because the vanadium seems to have been extracted in a higher extent in Earth gravity, while for phosphorus is the other way around. So in other words, it seems like the gravity has a different effect on different elements and on different microorganisms. So this is something that we have to take into account if we want to perform biomining in space. Nevertheless, the bioasteroid results allow us to demonstrate for the first time the microbial interaction with the meteorite material on the space station. And second, it also provided the first evidence of fungal extraction of elements from meteoritic materials in space. So we are quite satisfied about that. But what am I focusing on? I mentioned like the whole title of this seminar was about biorecycling. So why is the title about that? I'm getting there. These are, again, the potential roles the microorganism can perform for us beyond Earth. So, for instance, on an extraterrestrial settlement. Well, now I'm becoming more and more interested in this. Again, and this is as the aim of supporting life support system and its resource utilization. So recently, I've becoming more and more interested in the potential capacity of microorganisms of recycling waste in space. And why is that? Well, first of all, because if we can recycle and reuse materials, so in other way, close the loop in a space, achieving what is called circular space economy, then we can enhance the sustainability, the self-sustainability and the sustainability itself of the human presence in space. And this is going to be important not only to minimize the resupply of resources from Earth. In other words, the more you recycle, the less you have to bring with you during the space travel, but also if you think about that for ethical consideration that are associated with the space waste generated by the human presence. So which is the current waste disposal in space? I have a video here, I hope it works, that just show you what happens currently on the International Space Station to the waste. Just a very few amount of waste is currently recycled in space, and these include water and air. But all the rest is actually packed and then is ejected from the space station and is launched back to Earth where it burns in the atmosphere. So it's something that from the perspective of recycling, of course, is not very sustainable, but it's something that it makes sense when we think about an orbit that surrounds Earth. But if you think about going to Mars, to Moon or Mars or anywhere else, of course, this is not a system that is going to be viable and also sustainable. So we have to find a better way for long-term exploration. Maybe we can go on. Okay. And this is not new. In fact, space agencies are aware of the problem. They are aware of the fact that we are going to need to recycle waste as much as possible to achieve long-term human space exploration. And there are a lot of projects, particularly I always like to mention the Melissa project from the European Space Agency here, which try to understand how we can recycle biological waste and achieve, as you can see here, a circular economy, a closed loop system in which we can enhance the circularity and space waste management. This may sound familiar to you if you heard the talk from Dr. Marta Cortesao from June, which also mentioned a lot the importance of recycling waste in space, and particularly biological waste. There is, though, a lack in the understanding of what we can do with synthetic waste. What I mean with synthetic waste is metallic structures, electronic devices, clothes, and consumables. What are we going to that? There is at the moment, less research on this because it's harder to recycle. It's very hard already on Earth imaging in space. My perspective is that microorganisms could perform biological and synthetic waste recycling, but as we see so far, microorganisms respond unpredictably to the space conditions, and they also have deleterious effect on materials and crew. So in other words, research is needed. And this is what I'm focusing my current research. In fact, the objective of the research in the Space Microbiology group at the UK Centre for Astrobiology here in Edinburgh is focusing on unveiling the novel mechanism for microbial biorecycling and upcycle of synthetic waste, revealing the effects of space condition on this mechanism, and eventually also trying to understand how in the enhancement of this technology can support Earth and space sustainability. So both of them works hand by hand. So far, I am focusing on some, well, I am using different approaches, but I wanted to show you this one. This is a field work that I have performed in, 1 year ago now, on the northwest. So Edinburgh is here in Scotland, so north part of the United Kingdom. We went to the northwest part of the United Kingdom, so here in Scotland, and we collected some waste, synthetic waste, to understand if we can isolate some microorganisms of interest from this and then use them from the space perspective. But of course, there is much more than we can do from this. And what is the idea regarding the fact of synthetic waste biorecycling? To me, it is a promising tool toward a circular economy in space. You can see here from this image that waste recycling can, in future, become a circular compartment of a closed-loop system in which we have not only microbial biotechnology that are going to support space exploration in a sustainable way, producing recycling waste from different compartments and producing something that is useful for the compartment. So again, trying and getting loop closure as much as possible. But there is another point that to me is quite important for this perspective, and is that once we are going to have a very, very efficient system for synthetic waste biorecycling in space, then maybe the idea is that we can transfer these results to Earth so that we can address crucial environmental issues. And this is something that I am becoming more and more passionate about, because you may be aware of that, but the history of space exploration and space science in general, so I would say space exploration, space science as a history of producing cutting-edge scientific and technological advances that are first applied to space science, but then when they are developed, they benefit Earth. Think about GPS. GPS was originally developed just for space exploration, for space satellites, and now we have them in every electronic device, in every smartphone. We use them every day. Medical devices that have been developed for the astronauts' health, they are now used for medical devices here on Earth, and so on and so on. So there are a lot of benefits, terrestrial benefits for space science. And so I don't see why space biotechnology could not benefit Earth as well. But we have to study this, and we have to try and understand this. So in other words, if what I would like for you to take home, the take-home messages that I would like for you to have from this seminar is that long-term and long-distance deep space exploration will require sustainable approaches for resources and resupply, and microbes can really help us from this perspective. Finally, space biotechnology can enhance the sustainability not only of space exploration, but eventually they could also be transferred to Earth to tackle compelling environmental issues, but not much is known, and so we have to research. In the last couple of minutes of this talk, I would like to tell you just something about my adventure in the world of astrobiology, just because I don't know, hopefully this is useful, not because it's particularly interesting, but just because it may be useful for some of you, maybe. Having an example of how did I get to do astrobiology and space microbiology. I have to say that since I was a child, I was very passionate about science, every type of science, physics, astrophysics, biology, a lot of different type of science, any science really. But then the spark towards space came to me when I was 8, because my teacher organized an event with an astronomer that came in our classroom, and basically built a little planetarium just inside our classroom and showed us the constellations. And then they also organized a night out when we observed Saturn and the moon with the telescopes, etc. So basically at 8, I decided, okay, I want to become an astronaut. That was my goal at the time. But eventually just I really want to in future to work in space. And I also had another spark, and it was that this guy here, the astronaut Umberto Guidoni, which is the first European astronaut on board in the International Space Station, was actually living in the building, in the same building where I was living with my parents. So I managed to see him, I don't know, throwing the trash every day or taking the car, you know. I managed to see him every day, but I never approached him because I was too shy. But of course, the fact to me, imagine an 8-year-old child that was like, oh my God, there is an astronaut in my building. Oh my God, that's so exciting. He's been in space and all this kind of thing. So to me, all of this was really pushing me toward space science. Eventually, later, I went to high school and I started taking biology classes and lectures. And so this is where I developed a huge passion for biology. So I never grow out of my passion for space, but I became more focused on biology. And this is why when I get to the university, I decided to go I've done biotechnology as a bachelor at the University of Tor Vergata in Rome. And then I did my master again in biotechnology at the University of Rome La Sapienza. And then again, my PhD was in molecular biology, again on at the Sapienza University of Rome. And I also had the chance to go during my PhD. I went for a research period in Mexico and also in France. So this was my whole, training. But then when I was finishing my PhD, there was this thing of space back. So I was still passionate about biology, but this idea of space science was back in my head, and I was trying to decide what I wanted to do after my PhD. And I went to this event in which I met this guy again, which is the astronaut Umberto Guidoni. You may recognize him from the pictures before. And this time, as you can see, I approached him finally after years of never approaching him. And he gave a fantastic talk about how he managed to become an astronaut. And it was just about, okay, let's try. I love space. He was a physicist. Let's just try and go there. And I said, you know what, I'm going to try. And I realized that I love space. I love biology. There was this field, the astrobiology, that was making the two things together. And so I started taking a class, an online course in Coursera from Professor Charles Coquelle in Edinburgh, which is a fantastic scientist and person, I should say. And I started reading a little bit his work. He is very eclectic and does a different type of things. And I said, oh my God, I want to work in Edinburgh. I want to work in his group. And I was quite lucky because in that period in which I was trying to understand what to do after my PhD, a position for a microbiologist opened in Charles Coquelle group. So I tried to apply a postdoc position. I tried to apply and I got the position. And this is how basically I managed to work at the UK Centre for Astrobiology, becoming a postdoc there. And now my career, I have to say, is growing here in Edinburgh. That said, I don't have a cool conclusion for that. The only thing I would like to say is that astrobiology is a very, very interdisciplinary field. Yes, it is astrobiology, but in astrobiology, we have physicists, we have astrophysicists, we have biologists, geologists, engineers. So it doesn't matter what your background is. There is surely a way to get into astrobiology if you are passionate about it. And that said, I would like to conclude thanking all the people that were involved in my research so far. And also, these are the people from the UK Centre for Astrobiology, so my group. And these are all the collaborators that supported us and collaborated with us with all the results that I showed you so far. And the last thing I want to mention is that if you are interested about that, there is a new master in astrobiology and planetary science that we launched this September. So now, basically last week, it started here at the University of Edinburgh. So if you are interested, just read this QR code and see what is that. That said, I think I spoke more than I needed to. So I'm just going to thank you for your attention. I will be very happy to take your questions. And I'm going to...
So thank you, Rosa, so much for your fascinating lecture. We have a couple of questions. Okay, so you should be able to see the first one, and if you can read it, first of all, and then comment or answer.
Respond. Yes, of course. So, hello, there's already a rich database of plastic degraded fungi, so it can be another useful property of these organisms that can be utilized in the process of space exploration. Yes, thank you very much. I am working on that. I am working on plastic biodegradation and electronic waste degradation, and more generally speaking, electronic waste biodegradation is kind of a link between my previous research on biomining, because the very similar mechanisms that rule biomining are also applied to electronic devices, electronic waste recycling. So it's kind of something in between. But thanks for the suggestion. Yeah, there are a lot of plastic degraded fungi, and not only, even bacteria that that I'm focusing on in my future research. I hope I can give you more results as soon as possible on this. And then Thomas says, what is the most reliable bacterial strain used for bioproduction mining in space, or which microbes are best studied so far? Yes, so this is a good question, because the most, the bacteria that is mostly used in every biomining experiment or research that you will find in the literature, if you just Google biomining or bacterial mining, you will find Acidobacillus, which is a chemolithotrophic microorganism, meaning that it does not necessarily require organic material to grow, but it will require minerals and oxygen and so on. And in some ways, it's also considered an extremophile. To my knowledge, it's not been studied on the International Space Station, but I know that there is people that are studying them under simulated space simulated condition here on Earth, to understand if they can perform biomining under space conditions. I think, I may be wrong, but I think one of the reasons why it's not been studied in space, in real space yet, is because the conditions for them to grow are a little harsh. It grows at a very, very low pH. So it's not impossible. There are ways in which you can perform experiments with yet not trivial conditions like low pH or, I don't know, extreme conditions on the International Space Station, but it requires some levels of consideration when you design the experiment. And this is one of the main, I won't say limitation, but I would say it's something that, yeah, maybe limitation is the good word. It limits a little bit the science that you can actually do in space, because if you are in your lab, in your bench, you can. I won't say that you can do anything, but it's much easier to do a lot of stuff. But when you are in space, you have to consider, first of all, the health crew. So you don't want to. Yeah, you have to consider many, many different things. You want to consider how you are going to store the samples between the end of the experiment and the moment in which you are going to collect them and start analyzing them. You want to consider the facilities that are available in space. There is many, many things. I'm not sure if I answered your question. I hope so. If not, most reliable bacterial strain used for bioproduction. And the most reliable bacterial strain for mining in space and bioproduction, bioproduction and mining, I would say there are two different things, because it's the thing that you study, the results that you want to have, and the thing that you want to produce. It's really depending on, so you may use different strains, different bacteria, different microbes, depending on what is your application. So yeah, there is not a single answer, I think. Do you have any data which explain why there is a difference in efficiency of extraction? Is it more physiological or structural reason? I'm not sure what you mean with structural reason, but
yeah, I mean that because it's my question, so
yeah.
I can. No, I mean a structural reason is mostly defined by me that there may be changes in organization of, for example, fungi or cells shape, and that maybe this is that microgravity or other types of gravity can influence on the shape of cells, and because of that efficiency is lower. Or maybe it's just something physiological that, for example, I don't know, maybe expression of some proteins and is lower. So this is mostly this question.
No, that's a good question. The quick answer is that we don't really know. Of course, it's something that we are trying to address by mostly by multi-omic approaches. So by multi-omic approaches, I mean metagenomics, transcriptomics. Well, metagenomics will be more from the fieldwork, but transcriptomic, metabolomics, that will give us an indication of, okay, when the samples were in space, they produced these metabolites, and so this is potentially why they were producing these metabolites, and this may tell us something about that. But from the data that we have so far, there is not a clear pattern. So we really believe that it's really a not trivial question to address, because it may depend on the bacteria, or more generally speaking, the microbe that you use. It may depend on the rock that you're using, the mineralogy of the rock, and then the condition, not just the gravity condition, but even the growth condition. So it really looks like it's such, how can I say, it's such a complex system that is very hard to find the single. For instance, it would be very nice if I could tell you, oh yes, this gene is enhanced in the expression of this gene is enhanced in microgravity, so we know that that one is the one that is allowing microgravity in space, or not, or the contrary. The reality is that it's so complex that it's very hard to give you an answer, but this is why we need research from this perspective.
Sure. Thank you.
No, absolutely. Thank you. How did you search the meteorite? Oh, we just bought them. So there is a lot of companies, or even just collectors that whose either job or passion is to collect meteorites. And in both cases, you can just go to their website or just contact them directly, and they're going to provide you this. Of course, in our case, I forgot the name of the company. Oh, so sorry. I would like to quote the company that we use, but I forgot. But anyway, if you want to use that for an experiment, of course you want to be sure of what you are using. So in our case, we used a company that provides a certificate of exactly what that meteorite is, where it's been collected, when, and all these kind of things, so that we know for sure that we are dealing with elchondrite. I think it's called Skyfall, but I might be wrong, the company. Under what atmospheric condition were the biomining experiment conducted on the ISS? I assume it was not under vacuum, no. If not, then in a practical application, how will, say, an asteroid provide it? Yes, so the experiment, you're right, because the experiment was conducted inside the spacecraft, inside the space station. So it was at atmospheric pressure with the same atmospheric condition that the astronauts were experiencing. So exactly as it would be here on Earth. And it's a correct question, yours, because as of course, when you go on the moon or Mars, you're going to have different atmospheric condition. The reality is that either we manage to find microorganisms that, well, there is not much known about, there is a lot of experiments and studies on how microorganisms survive to vacuum. but not about how their growth in vacuum. So yeah, basically that. And it's because most of microorganisms that are resistant to vacuum and to low pressure, but particularly vacuum, they are resistant by forming by getting into a dormant state, for instance, spore. When they are in this dormant state, they are not metabolically active, so they won't perform biomining, for instance, or any application that you want them to perform. In other words, if in future we want to perform biomining on the moon or Mars or wherever, it is or the asteroids, it's very likely that we have to provide them a certain atmosphere, a certain liquid medium, or something like that. And in fact, there is an interesting research from Cyprien Verceau in Bremen, so in Germany. In his research, he tried to understand how we can lower the pressure constraint as much as possible so that there is more feasible, but not vacuum. If I'm not mistaken, there were more gravity levels studied other than ground condition and microgravity. Yes. Is there any trend for element extraction? Not really. There is a trend. I haven't showed you the results because it's still ongoing. So first of all, for BioRock, yes, there was more than one gravity condition. We tested Earth gravity, so microgravity, and then Martian gravity, and then Earth gravity. Well, actually, no, I want to... There is a trend from the perspective of... So it looks like higher gravities, very generally speaking, enhances the microgravity, the biomining capacity of the microbes. But again, it depends on which elements you are considering, which is the starting rock material, etc. So there is not one answer to this. It depends on many things. If we consider the first experiment, so BioRock, and you consider the rare earth elements results, then it looks like there is an increased trend toward the higher the gravity, the better they extract. But it's not statistically significant, first of all. So it's a trend, but not a real difference. And second, it's just for one of the bacteria from that rock for that element. If you consider another rock, another element, this may change. What is your opinion on future incorporation of synthetic biology and AI design of microorganisms for specific applications such as dedicated biorecycling of organic and synthetic products? So, design of microorganisms, I'm not sure if Hendrik means design, like using AI for instance, synthetic biology, so bioengineering, for instance, microorganisms to perform what we want them to do. Well, what is my opinion? Well, we have to see if it works. I don't have a negative opinion, or I don't have a positive opinion. It's just going to be. It would be quite interesting if we manage to have artificial intelligence helping us developing these things in an easier way, maybe in a faster way. What we saw so far is that, artificial intelligence in our everyday life and even in research is at its beginning. So there is still a huge need for not just a blind trust on artificial design. It needs to constantly be double-checked by the human brain. But that said, this is just the beginning. Who knows which are the capability in future? So no, I don't have a negative opinion. I don't have a positive opinion. I have just an open opinion, like I'm waiting to see what comes next. So this off topic, but if you had a very, very powerful spaceship and could take a flight about anywhere, where would you like to go? That's a very good question. So I would like to go outside the solar system, because when I was studying astrophysics, well, more astronomy, I was very fascinated by the I don't know, actually I studied this in Italian, so I hope the Oort cloud, Oort noob, I think it's called Oort cloud, which is this huge, massive, like sphere containing asteroids and comets that is outside the solar system. So this is where I would like to go. To do what? I don't know, but I just want to go there and be able to say, oh, that's very cool. But
sounds great.
Just to have fun. And then I could die happy.
Like a perfect place for holidays. So thank you so much, Rosa, for your lecture. I love your work in Edinburgh, and thank you to you all that you attended today's meeting, and follow our social media just to know when we are about our other activities of our society, and about our seminars. And also I have a small announcement that Polish Astrobiology Society organizes a conference which is gonna be from 1st to 3rd of December, and it's a life and space, so sounds like a perfect combination. 2023, so you can find the information on our website, and you're all invited for this conference. It's gonna be great.
Thank you so much.
Thank you.
Thank you. It was a pleasure.
Thank you.